The average drug was taking 12 to 13 years to develop. It was costing $2.7 billion to develop the average drug. And there was a 90% failure rate, much of that coming in late stage development after hundreds of millions of dollars had been spent on compounds.
Jeff is the executive chairman of Vertex Pharmaceuticals and the former CEO who led the most remarkable transformations in biotech. Under Jeff's leadership, Vertex went from a company that was looking to translate research and development into meaningful outcomes to repeatedly discover, repeatedly develop, and repeatedly commercialize breakthrough medicines.
We actually went back and spent 6 or 8 months, a group of us here at Vertex, and said, let's understand this history and understand what the problem really is before we try to solve it.
What are the biggest reasons innovation fails in biotech and pharma today?
This question of the innovation dilemma is so fascinating because you have to sort of start by looking at the data, saying, is there an innovation problem in this industry and others? And back in 2010, 2011, when we just started to look at the history of this thing, and there was clearly a big problem.
Jeff, I'm really excited to have you. I'll just quickly introduce you to the audience. For those that don't know who Jeff Leiden is, Jeff is the executive chairman of Vertex Pharmaceuticals and the former CEO who led the most remarkable, if not one of the most remarkable, but I'm going to say the most remarkable transformations in biotech. Under Jeff's leadership, Vertex went from a company that was looking to translate research and development into meaningful outcomes to one of the few biopharma companies to repeatedly discover, repeatedly develop, and repeatedly commercialize breakthrough medicines. Again, the company is well known for its work in cystic fibrosis, gene editing, pain, and has built a reputation for sustained innovation in an industry that, again, this sustained innovation is incredibly rare. And one of the exciting topics Jeff and I are going to discuss today is really around this concept of innovation dilemma and how Jeff's leadership has really created a culture of innovation that's truly sustainable. So, Jeff, before I pass the mic to you, you've spoken a lot about this concept. We've discussed this concept of the innovation dilemma. This idea that industries are investing more than ever in research and development, but again, they're producing fewer and fewer breakthrough products. From your perspective, what are the biggest reasons innovation fails in biotech and pharma today?
So first of all, Chase, thank you for having me. It's a pleasure to be here with you today. Thanks for the kind introduction. You raised the pressure on me a little bit, but thank you very much. And I can never listen to that kind of introduction without mentioning that none of what we did here at Vertex and the journey we're going to talk about today would have been possible without just an absolutely incredible team that I was able to recruit here back in 2011 and 12 and stayed with me and now with Reshma the entire time. And they deserve the credit. They did all the hard work, and all I had to do was provide a little guidance here and there. So I want to make sure to give a big shout out to them. So look, this question of the innovation dilemma is so fascinating, right? Because you have to sort of start by looking at the data, saying, is there really a problem? Is there an innovation problem in this industry and others? And back in 2010, 2011, when we just started to look at the history of this thing, and there was clearly a big problem. So I'll give you a few numbers just as examples. The average drug was taking 12 to 13 years to develop. It was costing $2.7 billion to develop the average drug. And there was a 90% failure rate, much of that coming in late stage development after hundreds of millions of dollars had been spent on compounds. It was very clear that model wasn't sustainable, and it was all moving in the wrong direction. Costs were going up, time was going up, success rates were going down. And so we started off by just saying, why is this happening? Because for 40 or 50 years, this industry had this incredible track record of producing transformative drugs, and it was slowing down, it was stopping, it was getting more expensive. And as we started to say, we clearly have to change that model. We can't succeed under those kinds of circumstances. First thing was to understand why it was happening. And so we actually went back and spent 6 or 8 months, a group of us here at Vertex, and said, let's understand this history and understand what the problem really is before we try to solve it. And we came up with 7 potential causes, or 7 contributory causes. There were probably more, but we thought that these 7 were the most important. The first one was that we asked, why do most drugs fail? And the sort of the bias in the industry at the time was, well, they fail because they're not safe, or they fail because they're not potent enough, or they're not hitting their target. That turned out not to be true. And by 2010, most drugs were failing because they were aimed at the wrong target, meaning they actually hit their target. They were potent, they were safe. But when you did that, the target wasn't the one that was the underlying cause of disease. And so the analogy I always use is it was like throwing darts in a dartboard match at the wrong dartboard. You could hit the bullseye 10 times, but you didn't score any points because you were aiming the darts at the wrong target. So the first problem was, how do we fix that? And we'll talk about that. The second thing, which was quite amazing, and it's true in other industries as well, was this lack of consistency and sort of approach, sort of commitment to a program. So what would happen in many big pharmas or biotechs, because these are hard problems, is people would work on them for 2 or 3 years and they wouldn't get anywhere. And they go, okay, we're going to stop working on cardiovascular disease, start working on respiratory disease. This isn't working. We're going to stop working on cystic fibrosis. We're going to start working on muscular dystrophy. But the thing is, it takes more than 2 or 3 years to solve these problems. And so this continuous movement in companies back and forth was a real problem. And it was made worse by the fact that you had all this leadership change at the top. The CEO of the company was gone in 3 years. The head of R&D was gone in 4 years. The new head of R&D had a new set of priorities. So everybody, ran over there and started working on that. It was sort of like the kids following the soccer ball all over the stadium. And so it was clear to us that, with these kinds of problems, you needed to work on them in a much more consistent, deliberate way for a longer period of time and understand there were going to be plenty of failures. The third problem, again, something that's also, I think, true in many industries is what I call the problem of organizational silos. So you had a research group and it was doing its thing over here. You had a development group in the clinic, they were doing their thing over here, a commercial group over here. And research would work on something for 5 years, create a molecule, and then hand it over to development. They go like, well, we can't develop this molecule. We're not interested in that. And then development would develop a molecule and they'd hand it to commercial and say, well, we can't sell this. So you had this lack of integration in product development cycles that was 10 years long. They were 10 years long. And that was a problem. So how do you get them to work together? And it was made a lot worse by these very long development timelines, because if it takes 12 years to develop a product and you only have 20 years of patent life, well, guess what? Even when you commercialize it, there's only 7 or 8 years left to recoup your investment. Big problem. Then there were some other things like scaling challenges, particularly in manufacturing. You often made drug at small scale for clinical trials. You had to scale up. That could take 2 or 3 years and tens of millions of dollars. And that slowed the whole process down. And then once you got there, you got into these other 2 problems, which were interesting. One was what I call the fast follower problem. So the fast follower problem was somebody works for 10 years to make a new class of drugs, a new statin, the first statin. And it's a pretty good drug, but it's not a perfect drug. And then they stop. And then everybody's following along. And pretty soon 3 other companies make fast followers and they put the first person who did all the work and investment out of business. And that happened to us at Vertex. The first drug at Vertex was this really novel molecule for treating hepatitis C. It was a real breakthrough. We worked on it for 12 years, doubled the cure rates in hepatitis C. The drug was the fastest drug ever to $1 billion. And then we got out-innovated literally a year later by Pharmaceuticals Gilead, and we went to 0. We actually had to take the drug off the market because we stopped innovating. So the notion of this stopping the innovation at the first molecule was just deadly for the industry, and we had to fix that. And then the final thing was this failure to integrate internal innovation, your own research efforts, with external innovation, which is business development. Right. So most companies had an internal research effort that was working on disease A, B, and C, and then they had a business development group that was going and licensing molecules for diseases X, Y, and Z. There was no integration. And then when they licensed something in, the internal team said, hey, we don't want that because it's going to take away from our budget. We don't want you to take away from our internal programs. So this disconnect between internal innovation and external innovation created these very weird dynamics of what was actually being developed. So those were 7 reasons that we saw that were just sort of deadly to the whole process.
Yeah, I mean, and I'm excited because I want to jump into each of these reasons more specifically, because obviously there's a lot to uncover there. My first question before we jump into the different reasons really centers around the moment of that epiphany, i.e. the moment that you realize the existing model just wasn't going to work. Was it a single moment? Was it a set of moments? I know at the time before you took over as CEO, you were on the board. What was that first epiphany, that first spark that led to this cascade of really, I'll refer to it as first principles thinking on, and again, others just want to follow the typical roadmap, but you really took a first principles approach towards understanding the basic problems at their most basic foundations.
I think the epiphanies really came from watching the industry, but also watching Vertex. Obviously, the success and failure of Incvac, the hepatitis C drug, that's an epiphany. When that happens to you in a company, that is a moment of crisis where everybody goes, "Uh-oh, what just happened? We got to do something different." That was part of it. Part of it was also watching how the industry was attacking this problem. It's not as if we were the only ones who understood there was a problem. The industry clearly understood there was a problem. But what was interesting to me, Chase, at that time is most of the industry's response to that problem was just start more programs. So in other words, if your failure rate is 90% and you only have 10 programs, you're only going to get one. So let's have 100 programs because then we can get 10.
More shots on goal. More shots on goal.
It's the more shots on goal approach, right? And literally people would talk about that, like just more shots on goal. That's all we need. And they spent a lot more money with more shots on goal. But the problem was it didn't scale. The failure rate kept going up and up and up. The expense went up and up and up, and they didn't end up with more drugs. So it was very clear. And by the way, that was true at Vertex too. We had 23 programs in different diseases at that time. And I was looking at that going like, no, that's just not going to work. That defocuses the entire organization. So those were sort of the epiphanies of like, A, it happened to us, and B, more shots on goal isn't going to work. So we better go back to first principles and come up with a way to increase the probability of success. That's the answer, and shorten the timelines. How do we do that?
Yeah. And again, one idea that you highlighted was this concept, and it ties to what we're discussing, which is that many drugs fail, again, because they're not failing necessarily because they're unsafe or they're poorly designed, but because companies are just pursuing the wrong biological targets. How did that insight shape how you as a leader at Vertex decided which diseases and targets should be pursued?
Yeah, it's a great question. And actually, in many ways, it was the fundamental change that we made at Vertex, which was we came up with the strategy that said we're only going to work on validated targets. Mm-hmm. And we defined validated in a very specific way. It had to be a target that was known from either human genetics or from pharmacology to be the underlying cause of the disease. So that we knew if you hit that target, you were going to fundamentally modify the course of disease or cure the disease. And the first example of that for us was cystic fibrosis, where we knew that the CFTR gene was the cause of the disease. Every patient had mutations in the CFTR gene. And we also understood the biochemical basis of that. That's the CFTR protein was a channel that caused ions and water to flow. And in these patients, they had a defective channel, so they couldn't hydrate their secretions. So we absolutely knew if we could repair the function of that defective channel, we would modify the course or cure or prevent the disease. And every disease that we've worked on since then, every single one has been a validated target. So we created this thing at Vertex called the sandbox. The sandbox was the place where our research group could play, sort of like your kids, right? They can play in the sandbox, they can't play outside of the sandbox. And we then curated a set of diseases. We now have 60 to 70 diseases that fit all of the criteria for a corporate strategy. And every one of them was had a validated target where we knew from genetics or pharmacology that if we hit that target, we would cure or modify the course of disease. And that's been just an absolute rule, both for our internal research and for our BD efforts. And as we'll talk about in a minute, that changed our probability of success from a 90% failure rate to an 85% success rate. That's just transformational in the whole economics and efficiency of the model.
And more specifically on the concept, on this key concept around Vertex's strategy and idea of the sandbox, which again, to me, just makes rational sense, right? It's funny because the obvious things are not so obvious until they're out in the world, is what I always like to say. How does this sandbox approach drive both scientific focus and then also strategic discipline within the organization?
Yeah, it's a great question. Let me answer the second part of it first, because to me, the discipline is what's really important here. Because I think many companies would tell you, well, we work on validated targets too. But then you see them do Alzheimer's trials, because they would say that beta amyloid is a validated target for Alzheimer's, but it's actually not. Yeah. There's data suggesting that beta amyloid is associated with the disease. That's different from being the causal biology of the disease. And so how many trials, I think there have been 15 or 17 late stage trials in Alzheimer's disease that have failed, costing billions and billions of dollars, not because the drugs weren't well engineered. They're actually very, very well engineered drugs, and they get to where you want to be, and they hit the beta amyloid target. It's just beta amyloid isn't the right target, right? So the discipline has to do with not letting yourself slip off of do we know it's the causal target, or do we think it might be the causal target? And you sort of start to move down this logic chain that says, well, but the evidence is pretty good that it's the causal. Let's just do it. And that's where I think companies sort of slide down that slippery slope. And at Vertex, we just don't do that. We have these very vigorous discussions about, do we know it's the target? Has human genetics proven it? Has pharmacology proven it? Or do we think it might be the target, or it's in the pathway for the target? That's different. So discipline turns out to be really important. And then we absolutely insist that we only work on this curated set of diseases that's decided in a very formal process all the way up to the top of the company. So to get into the sandbox, you need the chief scientific officer and the CEO to sign off on that disease in a very formal process. And you just can't work on diseases that aren't in the sandbox. And we've been pretty disciplined and tough on that.
Yeah. I'm curious if we were to get an inside scoop around how the decision comes to determining what goes into the sandbox and what goes out. Any more you can share there?
Oh, sure. So there's actually a group that includes the CEO. It still now includes myself. It includes the CSO, the head of development, the head of regulatory. It's the senior group of essentially scientists and physicians at the company. And by the way, just to mention, 70% of our employees are scientists or physicians. Another sort of change from what's true in most biotechs or pharma, certainly. But this senior group has a formal process where a disease has to be nominated for the sandbox. And there has to be a white paper written about why it's a sandbox disease. And by the way, it's not only that it's a validated target, it has to be potentially transformative, it has to be a serious disease, it has to be a specialty market. There's a set of criteria that we use. And someone who's going to nominate a disease or a group writes a white paper, it comes to this committee, it's actually discussed, and it's either approved or not approved. So there's a very formal process, and there's a slide, there's a list of all the sandbox diseases. So everybody in the company knows what those diseases are.
Interesting. Very interesting. And thanks for sharing that, Jeff. Now, the other thing you hit on is really this concept about creating tighter integration between different groups, right? Research, development, manufacturing, regulatory strategy, commercialization. How did Vertex operationalize that inside the company? And I'm curious, why do you think it matters so much for improving R&D productivity?
Yeah. Or I would just say sort of medicine productivity, right? Going all the way from R&D through to commercial. So we operationalized it two ways. One is at the top. So we have what we call an EC, an executive committee, that has the CEO, the CFO, the CSO, the head of development, the head of regulatory, all the relevant functions, the head of manufacturing. And that group meets or met with me and still meets with Reshma every week for 6 hours on Mondays.
Wow.
And they review all of the programs and the progress in the programs, and they help make all the tough decisions. For instance, all the decisions about when to progress a program from research to development to commercial. So the senior leaders are in the room. They're all signing off on this at the very beginning. So they're looking at the research programs, and if the head of commercial sees something in research and he or she says, hey, wait a minute, I don't see how we're going to sell that drug. I'm not enthusiastic about that. That matters. I mean, they can actually veto that program in that group. So there's a senior group that's cross-functional that's meeting every single week to discuss these programs. But then we cascaded that down through the organization. So every program that we have, even early research, has a representative from regulatory, from development, from commercial on that team from the very get-go. So we never get to a situation where research has produced something and then it's handed off to development. The development folks go like, well, wait a minute, we didn't know about that. We can't develop that. And so it makes a huge difference because there's none of this fumbled balls as we go through the process. And for instance, commercial helps define the profile of the compound when it's still in research. We have a target product profile that commercial has signed off, and it says the medicine has to look like this. It has to be once a day. It has to hit this target. It has to be tolerable like this. And so we have a profile from the very get-go that goes through the process. So we don't end up with those disconnects.
I have to say, Jeff, it's incredibly impressive, just given the size of the company now. You're now, what,
8,000 employees.
8,000 employees, $115 billion market cap company. Yet the way you're describing internal communication really sounds more like you're still operating at the startup level, which is truly incredible when you consider that size and scope. And I'm comparing that to the way that other big companies, not just within pharma, but outside of pharma, operate. So it's incredible that you've been able to maintain this culture of scientific breakthrough and innovation, which really takes me to my next question around, you and I, we've talked about this concept of serial innovation, right? How do you continuously improve therapies to stay ahead of the potential competitors, right? Which ultimately, by the way, becomes a benefit for HCPs and patients, right? I always like to say, I'm always wary of using the word competitor because competition actually is what provides greater benefit to patients and doctors. And that's why we're all in this industry. How does Vertex and how has Vertex built a culture that really encourages teams to keep innovating even after major scientific breakthroughs? Because what I've noticed is a lot of companies, they hit a scientific breakthrough, they have a blockbuster product, and you kind of reach this stage of complacency. How has Reshma, how have the two of you created a culture that centers around this concept of no complacency, continuous serial innovation? In fact, I feel like the acceleration goes up every time a breakthrough is discovered. And so help me understand that. It's incredible. And I know a lot of folks that listen to this are biotech CEOs and others, and I know they're trying to crack the code here.
We call it constructive paranoia, and it goes back to our Incivek experience, right? That was such a crushing experience. We were so excited about this new medicine that doubled the cure rates of HCV, and all these patients were using it. And then there was really this oh no moment when we realized, we stopped innovating. We just got out innovated, and this thing is gone. That was a very negative experience. But for the future of the company, it was actually a very positive experience. So it created this constructive paranoia on the one hand. But on the other hand, Chase, it's what you say, which is our feeling is the really hard part is cracking the biology. The first time you crack the biology on a program, making the first drug, that's really, really hard. Making Kalydeco, the first CF medicine, that took years, and it was just a really hard problem. But once you crack the biology, actually it's put the pedal to the metal, because now you've done that. You can do it again and again and make better and better products. And so in every program, we have this life cycle development sort of plan. And once we've cracked the initial biology, we actually put more resources on that program to do the next and the next and the next. And we always remind the teams of the competition, because everybody jumps on once you've had the first success. And we're like watching companies A, B, and C. They've now got a CF program. We better go faster. Where do we need to go next? And of course, the irony is that the first company has a tremendous advantage. You've spent all this time and money to develop the expertise around the disease. You are better than any competitor because you have the assays. You know what the drugs have to look like. You have the biologists inside. So the fact that those companies stop always seemed to make zero sense to me, because they actually had a competitive advantage. It's just that they went on to the next drug, and we just don't do that. So every program, if you look at CF, we have 5 medicines now, one better than the other and the other. If you look at pain, we have the first one, we have 6 coming behind it, and we're excited about taking those all into the clinic to see which one is best. If you look at our sickle cell program, first approved gene editing therapy, but now we're going to make for gentler conditioning, and then we're going to do in vivo gene editing. We have a whole plan, but we have this tremendous group of biologists and chemists that already have the expertise. So in a way, it's easier to accelerate than to stop.
Makes a lot of sense. And I'm happy you brought up pain, because that was one way I wanted to make this even more tangible. I.e., Vertex has spent years developing a new class, decades. Yeah. Developing this, new non-opioid pain medicines, right? And of course, we've talked about Journavx. What motivated the company to pursue pain as a major area of innovation? Because when I was talking to Reshma originally, and Reshma told me we're pursuing pain, and this is before I, of course, understood and had a deeper understanding, which again, I've had the privilege of getting to learn more about through you and Reshma and others around learning about the sandbox. But now it's, of course, obvious to me. Again, the obvious things are not so initially obvious. But what motivated the company to pursue pain as a major area of innovation? And out of curiosity, what has made this approach different from previous efforts that, again, have been pursued to develop non-opioid pain treatments?
Yeah, so pain started, by the way, back in 1999, if you can believe. We've been working on it since then. And it had the two classic features of a disease or three classic features of a disease that fits the Vertex, right? First, very serious disease, very large unmet need, right? We hadn't had a new pain medicine since really the 1960s with NSAIDs and et cetera. So very large unmet need, and a very important disease, obviously. There are 100 million opioid prescriptions written in the U.S. every year, which is pretty amazing. Second, obviously the opioid crisis, right? We had a drug which was very, very effective. Opioids are very, very effective, but very, very dangerous. Bad side effects and obviously addiction in up to 10% of the people who take them. So that was another part of the need, right? And then the next part was the validated targets. So in the late 1990s, there was this experiment of nature, a genetic, but a really experiment of nature, where these three Pakistani families were identified who had what was called congenital insensitivity to pain. So the initial family was amazing. It was a 10-year-old kid who was a street performer in Pakistan, and he would go out and perform and put needles through his arm and feel no pain. He could walk on hot coals and feel no pain. He did it actually for a living. It was sort of remarkable. And then these scientists identified three such families, and turns out all of them had mutations in a single class of genes, these NAV sodium channels. And so we had the validated target, if you will. That is an experiment of nature that tells you that you have a validated target. We knew if we could hit that target, we would fundamentally alter pain perception. So we started in 1999. And we kept going in 2005, making no progress. And we kept going in 2010 and made no progress. We had a fantastic team working on this. We had the channel identified, purified, we had assays, we had everything. We could not make a drug that would hit this channel the way we needed to hit it. And we went on to 2015. And one of the pivotal moments in 2015 is I had a great CFO here at Vertex who was my partner. And he sort of looked at us, he came into my office one day and goes like, Jeff, this is ridiculous. We've been working on this for 16 years. We spent tens of millions of dollars. Enough is enough. We're just not going to be able to make a medicine. And I sort of said like, yeah, maybe he's got something. So I called up the head of the program in San Diego, a fantastic scientist named Paul Negulescu. Paul got on a plane like that day, flew out to see me, and he goes, Jeff, I think we're making progress. We have a new idea how to do this. Please just continue funding for 2 more years, and I think we're going to get there. And if we don't, okay, enough is enough. And sure enough, we had this young scientist in San Diego who had made a very clever observation about a different way to hit this channel in 2015. And within a year and a half, we had a whole portfolio of novel kinds of molecules, the first of which is a NaV1.8 inhibitor called Journavx. And as you said, that was just approved a year ago. And it basically has opioid-like efficacy with no addictive potential and none of the side effects of opioids. And now we have a whole portfolio of these. This will really revolutionize the treatment of pain.
And for the listeners, just to really paint a clear picture of exactly how this product works, the analogy I thought of before this, because I always love to use analogies, is opioids work by, think about it as turning down the pain processing in the brain, right?
Correct.
Whereas Journavx works by actually cutting the pain signal on the road before it gets to the brain, right?
Perfect explanation.
And again, and Jeff, I know you discussed how it works with the sodium channels. Obviously, again, we have pain sensing nerves, like this individual, the street performer in Pakistan that was putting needles through his arm. Again, we have these pain sensing nerves that go throughout our body. These pain sensing nerves use tiny proteins or wires called sodium channels to send pain signals. And again, as Jeff is explaining, Journavx blocks a specific one, NAV, which is found mainly in the peripheral nervous system. So it's it's really cool when we think about the again, being able to, it's kind of like you're stopping something, you're stopping something more upstream before it gets to the source, which is really incredible. Jeff, I'm curious if this new class of pain medicines, which again, I'm very excited about. I think every single HCP should be beyond enthusiastic that there is now a real alternative to opiates. If this new class of pain meds delivers on its promise, how can this change the way physicians treat pain and how patients experience pain management?
Yeah, it's a great question. So Journavx was approved for acute pain. So think about post-surgical pain, think about injury pain, accident pain, those sorts of things. And it's already really changing that in several ways. So if you think about it, for anything that was severe acute pain, like post-surgical pain, in many ways, generic opioids were the first medicine that many physicians tried because they knew they needed the efficacy, right? So they were almost upfront medicines. Sometimes they try NSAIDs or Tylenol, but usually that didn't work. So opioids, they very quickly reached for opiates because they had to provide some kind of pain relief. This sort of changes that paradigm, right? So now clearly, before you go to opioids, you want to use a drug like Journavx because it doesn't have the addiction potential and it doesn't have the side effects. Maybe another way of saying it is opioids become the last alternative instead of the first alternative. That's a pretty fundamental change. But we're only at the beginning of it, Chase. If you think about it, because as you said, this is a NaV1.8 inhibitor. There's another channel called NaV1.7, which is also involved in those wires in the periphery. We now have NaV1.7 inhibitors. There's a lot of evidence that we and others have accumulated that if you inhibit both NaV1.7 and NaV1.8, you get even more profound pain relief. And again, no addictive potential. So the next plan is let's combine our NaV1.7s with our NaV1.8 so we can get even more robust pain relief. And in fact, one of the things that we're going to have to start thinking about is sort of a good problem to have is in chronic pain, in low back pain, in chronic knee pain, et cetera, where we're testing these drugs and hopefully we'll get them approved there too. We have to be a little careful that we don't provide too much pain relief, right? Because if you have a bad knee and you're having pain, well, that's a signal to you. You better stop playing tennis on your bad knee when it starts hurting. And if you have pain medicines that are too potent and they can prevent that pain, you can actually inadvertently produce injuries. So we need to dial the pain dial down to the right point, but still have some perception of pain. You don't want to be able to put your hand on a burner and not feel it. And so part of the trick here is finding the right combination at the right dosage levels to give you really potent pain relief, but you still have some acute pain feeling when you need it.
Absolutely. And, again, it's really a balance, right? It's really about finding that balance. I want to tie back this conversation around the pain program back to the broader strategy that you and the team have built at Vertex. Again, including the sandbox model and the focus on validated targets in transformative medicines. Looking ahead, where do you think the biggest breakthroughs in medicine will come over the next decade?
Yeah, so let's take at a 50,000 foot level, right? The two most important things that have really changed over the last 15 years, now I guess it's more like 25 years. First, the sequencing of the human genome, right? Because prior to that, the top 100 drugs were made against only 400 targets. We know there are 20,000 genes, proteins, at least 20,000 targets in the human genome, and yet the top 100 drugs have been made against 400 targets. So the sequencing of the human genome just rapidly expanded our target base. We now know most of the potential targets, if not all the potential targets. And the trick is just lining up the right target with the right disease. But that's huge once you know what all the targets are. The second thing that's changed is the number of therapeutic modalities, right? 50 years ago, everything was a small molecule, it was a pill, an organic chemical. Then we went on to protein therapeutics and monoclonal antibody therapeutics. Really expanded the range of targets we could approach. Now we're going to a whole new set of targets, cell therapies for regeneration. We'll talk about that in diabetes, and genetic therapies where we can actually alter the human genome or affect the human RNA. So genetic therapies that actually can target very specifically given genes, mRNAs or proteins, that's a whole new therapeutic modality. So when you combine now we know all the targets, and now we have all these new therapeutic modalities. You put those together, suddenly you've just opened up the spectrum of diseases that you can treat. And the first example I would give you, of course, it happens to be a Vertex example, is the human genome editing for sickle cell disease, right? Yep. Sickle cell disease was the first molecular disease. I mean, Linus Pauling, in an amazing paper in 1948, described the genetic and biochemical basis of sickle cell disease in 1948. It was incredible. And he showed that it was a mutation of the beta globin gene that caused this very peculiar aggregation of the protein and sickling of the cells, et cetera. He just explained the disease. It was really an incredible paper. Yet, despite that, we had no good treatment for sickle cell disease until 2-3 years ago, because we just didn't know how to reverse that problem. And if you tried it with small molecules, and companies did try it, so far it was impossible to make a small molecule that would reverse this biochemical defect in the beta globin protein. That all changed with the advent of human gene editing, right? In 2012, once you knew that you could, that was described by Charpentier and Jennifer Doudna, they described a way you could actually go in and specifically surgically alter the human genome at one base pair or one site out of the 3 billion sites. The minute you had that scientific breakthrough, new therapeutic modality, you had a new way to potentially treat sickle cell disease. And so, as you know, we partnered with CRISPR Therapeutics in 2015. We took hematopoietic stem cells, CD34 cells, out of the body of these patients with sickle cell disease, put them in a dish, used CRISPR-Cas9 to edit this one site in the genome, make a single cut that then turned on fetal hemoglobin, put those cells back into the patient where they home to the bone marrow. We knew they would from bone marrow transplant, and essentially have cured more than 90% of the patients that we treat with a single treatment. There were two things there, right? One was the molecular definition of the disease, 1948. But the other one then was this new therapeutic modality, 2012, just opened the door to treat this disease. And by the way, that of course opens the door to treat many genetic diseases, because there are as many as 7,000 genetic diseases for which we now are understanding the single gene or genetic cause. And once we get better and better at editing, particularly editing in vivo in the body as opposed to ex vivo, it opens up a possibility to treat all those diseases. So that's one example. Type 1 diabetes, we're also working very hard, as are others. Again, a completely different therapeutic modality. The description of a human stem cell that could be embryonic stem cell that could be differentiated into different cell types in the last 10 years, 11 years, another Nobel Prize, opens up a whole new possibility. Now we could take stem cells from a patient or even from a normal person, and we can turn them into lots of different cell types, including islet cells, beta cells from the pancreas. And we know that patients with type 1 diabetes have destroyed their own beta cells. That's why they don't make enough insulin. That's why they need to get exogenous insulin. And until now, the only treatment was daily injection, sometimes multiple times of exogenous insulin. And it wasn't very good at controlling the disease. But it was pretty obvious if you could make beta cells and put them back into the person, that would essentially cure the disease. Couldn't have a better validated target than that. The problem is we couldn't make them until Doug Melton came along, worked 20 years to figure out how you make a stem cell into a beta cell. We then bought his company, Sema. We turned that into a drug process where you could actually manufacture hundreds of billions of cells, put them back into patients. I think we've reported on 14 patients that we've treated single infusion of these stem cell derived beta cells, and 10 of the 12 are off of all their insulin. They have normal glucose control. So it's essentially cured their disease. So again, think about that for other places. Think about it for the heart. Think about it for the brain. Think about it for joints. Regenerative medicine using stem cell derived cell types is going to be a whole new therapeutic modality, and it's going to revolutionize the treatment of this huge range of organ diseases and organ failures.
How do you think about it? It's clear that there is a really robust system that's in place around how you're finding those experts within science who have identified those validated targets, and you're able to work very, very quickly on confirming that validated target, and then working quickly on creating cures. I'm going to throw one out there that is a very challenging disease where we haven't found, and I'm just curious. I want to get your thoughts. And if you can't answer the question, Jeff, I understand. But I want to choose a disease like ALS that is incredibly challenging. We still don't really understand. We have all these different theories around what could be resulting in ALS, genetic causes, non-genetic causes of disease. How do we find the validated target? I mean, even Alzheimer's is another example that you were hitting on earlier, where we've always thought it all comes down to the alpha beta plaques, the beta amyloid. How do we find a validated target? Is it an investment more in basic research? Is it through partnership? And again, if you can't, if I'm just curious, I have to get your thoughts on this.
Yeah, so I don't have a magic answer for you or a simple answer, but I'll give you a couple of thoughts maybe about it.
Please.
And ALS to me, both the diseases you mentioned are perfect because they sit right outside the sandbox for us. In other words, they have all the qualities of a disease we want to treat. It's a serious disease. It's a specialty disease. Everything that we want. And we can make a transformative therapy, except we don't have the validated target.
Yeah.
So, and by the way, diabetes used to type 1, sat right outside the sandbox. And we knew that all we needed to know is, could you make a stem cell into a beta cell? How do you make beta cells? And the minute that David Altshuler and I knew Doug Melton very well for many years, and we followed SEMA. And I still remember the day when he came over to the conference room and he pulled a little vial out of his pocket and he said, guess what? We solved the problem. We made beta cells. And he showed us data. We bought his company 3 months later. Wow. So we knew that disease moved into the sandbox like the next day because we had what we actually needed. And ALS sits out right there too. As soon as we have a validated target, amyloid sits there too. So your question is the right one. How do you identify it?
Yeah.
And it's going to be a little bit different for different diseases. I'd give you 2 thoughts. I think there are some diseases, and I sort of suspect Alzheimer's is one of them, where there will be maybe not one, but several, not hundreds of targets that you have to go after. Alzheimer's isn't going to be a disease, I think, for most people that has this very complex, hundreds of different genes contributing to it, although there will be other diseases like that. I think there will be not one, but a few. And those diseases will fall. I mean, they'll fall soon, I think, because as our understanding of the interactions between the human proteome get better and better, and our understanding of pathology gets better and better, we will identify those. And there's some candidates already, right? Alzheimer's, the tau protein seems to play a very important role, not in everybody, but in many patients. ALS, I think, is going to be the same. We know there's a small fraction of ALS that is genetic. That's usually a pretty good clue as to the pathway. But we know that those genes aren't involved in the majority of patients. But it's probably in that pathway. The better we understand that pathway, the more we'll be able to divide patients into 2 groups, 3 groups, 5 groups of these have this target, these have this target, these have this target, and then we'll be able to come up with the answer. So I'm very optimistic about those kinds of diseases. I think there are other diseases that are going to be more complex with very complex genetics and many different ways of getting there. Certainly the fibrotic diseases may be that way. It may be harder to come up with simple targets. But, in CF, what happened was interesting because we knew there were 140 different mutations in the CFTR gene. And initially we thought, we're going to have to make 140 different medicines potentially. But it turned out that as we understood the biochemistry better and better, we could actually divide it into just 2 or 3 groups of patients with those 140 mutations. And then we could make 2 or 3 different medicines that hit those groups, and we solved the problem. And I think that's what's going to happen in some of these diseases. So I'm pretty optimistic. I think the hardest one, Chase, is cancer. And the reason is because although there are definitely even single causative mutations that lead to cancer, once you have cancer, you get this genetic instability and you get this continuous new set of mutations around your therapies. And I think that's particularly challenging. But again, new therapeutic modalities may enable us to get around that. Things like CAR T cells and immunomodulatory therapies that can sort of adapt with the patient's immune system can adapt to the new mutation. So I'm not hopeless about those diseases, but they're just a little harder.
And again, Jeff, there's so much I want to discuss with you. I know that we're coming closer. We don't have too much more time to get into. I have a lot of questions about your thoughts on epigenetic reprogramming and things like that, but we'll save that topic for some other time. I have to move on to commercialization because at the end of the day, no matter how much incredible R&D goes into the development of new therapeutics, if patients and HCPs don't know they exist, then from the eyes of the patient and the doctor, they simply don't exist. And so how do you think about, we've talked about a lot about, and you've argued that innovation really needs to happen across the entire value chain, not just in the lab. So what do you think the next generation of biotech commercialization will look like? Out of curiosity.
Yeah, it's a complicated question because it's different for different medicines. We'll talk about that in a second. But fundamentally, I always think about, I'm a sort of simple guy. So I always think about commercialization as having two important components. One is create awareness and demand, which is what you're talking about. How do you make physicians and patients aware that the medicine is there and want to get it for their disease, appropriately so when it's indicated? And then reimbursement, right? You have to have the financial incentives. And those two have to go hand in hand. Let's talk about demand first. So with respect to demand, we've been very fortunate because in many of the diseases, in many of the very serious diseases, they're very scientifically driven with respect to demand. So let's take CF as like the poster child. All the patients with CF in the United States, all 35,000, are treated at 270 centers by the same group of physicians. They are all, the CF Foundation provides continuous streams of information to those patients and their families. Those patients would know about one of our medicines when it entered phase 1. I mean, long before it was approved, and they were waiting for it. We'd get letters like, when's Trikafta going to be approved? How far away are you? They knew the day. We didn't need sales reps out there selling it or DTC advertising or anything else. The day it was approved, every patient in the country who had mutations, they pushed a button, the patients came out, the CF center scheduled them, and 90% of the patients were on drug within 6 months. So that's one end of the spectrum, right, where it's just a pure scientific sell. And I think there's some lessons to be learned from that, because I think the more that patients and patient groups can mobilize themselves, the better, the more quickly they'll get access to and become aware of drugs. And one of the things we're trying to do is to leverage that in some of the other diseases. The sickle cell community is not nearly as organized as the CF community was for a whole variety of reasons. They should be, right? We want to get them organized. In the pain world, right, there are whole organizations out there of families and moms who unfortunately lost a child to an opioid overdose, right? That is a natural venue for creating awareness about Journavx, because those folks are really invested in making sure that doesn't happen to somebody else's child. And we need to figure out how do we work with them to help create awareness in that community. So I think patient communities are going to become much more important in terms of awareness. I'm hoping physician communities can become more important as well. So on the demand side, I think it's a combination, depending on the medicine, of what you're going to do, but you need awareness and you need physicians aware. And I think the old model of, send 5,000 sales reps out there to see physicians, every week, that's not how it's going to work. We need to leverage information technology, AI, and patient communities much better.
Agreed.
On the reimbursement side, it's more complicated because I think, unfortunately, we and somewhat inadvertently sometimes, we've created disincentives. We've created disconnected incentives between insurers and payers and patients. In other words, pharmacies, insurers, and payers often make more money by prescribing generic than they do by prescribing a new branded medicine that's better, or prescribing a branded medicine that's not as good that's cheaper instead of one that's better because it's more expensive. Those are really weird, perverse incentives, and we have to do away with those. At the end of the day, patients should have access to the best medicines, not just the medicines where a pharmacy makes more money or a physician makes more money or an insurer makes more money. That's a bad incentive. And that's just, I don't blame them for going where the incentive is. We have to change the incentive system so they're incented to actually get the best medicine. One way of doing that is to create more compelling health economic models. One of the problems with our system is we don't really envision the entire health economic model of a new treatment. So when one of our CF medicines, or let's take our sickle cell medicine has a list price of about $2.1 million for a single treatment. Wow, that seems like an incredible amount of money. Why would anybody want to pay that kind of money? How do you incent people to do it? But actually, you can put together a very convincing health economic argument where you say, if we cure 93% of the patients and they never need to go in the hospital again and they go back to work and they live a long life, what are the economic consequences of that compared to the 2.1 million? By the way, they're incredibly good. It makes a whole lot more sense to pay the 2.1 million than the millions and millions you're going to pay for years of hospitalizations and lost work and productivity, et cetera. But our system isn't set up that way. It doesn't look at it that way. The insurer just sees the upfront payment. The system doesn't see all the gains in productivity because somebody else is paying for those in all sorts of different ways down the road. We need to do a much better holistic model of looking at the cost and benefit of these drugs financially, because that will allow us to put in place aligned incentive systems and really value these drugs properly. Right now, they're not valued properly because we have this strange, disaggregated incentive system.
Yeah, and if you think about it, the analogy that was going through my mind as you were talking through the sickle cell treatment is, we want to incentivize the right behaviors through health economics, right? You look at a treatment like Vertex's sickle cell treatment, right? It's designed as a one-time treatment. I know the label says it's the FDA labeled it as it was a one-time single dose intravenous use only, I believe. I was looking at it before our call, so I might be slightly off. I apologize if I was slightly off. But again, if you consider the incentive system, it's like asking, I love to think in analogies, Jeff. I know you're patient with them, but it's like tire companies, right? If you had the ability to create a tire that never needs to be replaced again, right? If the incentive was there to do it, then we'd probably have indestructible tires, right? And so if you have the ability to deliver a one-time treatment to a patient where they don't have to repeatedly receive follow-up treatments, and you add up the cost of those follow-up treatments, you add up the cost of all the surrounding complications that come with sickle cell pain syndromes and all these other unfortunate consequences of disease that we tend to see around sickle cell. To your point, not only are you providing a significant greater benefit to the patient, but yeah, as a whole, there is a very clear health economics argument for it. And I just, maybe this is me just being too aspirational. And, again, hopefully I'm planning on spending my entire life in this industry. So the plan is to do what we can to hopefully change things for the better and creating really the right incentives to continue to provide the right support to companies like Vertex that are making these big investments in creating these curative treatments. So again, my hope is for those of you listening that are in the health economic sphere, I know we've had some folks from HHS and public health leaders that do listen to this. Please, we all need to work together on creating the right frameworks to continue to ensure that companies like Vertex are doing the work that they can to keep curing many of these diseases, because it's critical for us as HCPs and it's critical for every single patient out there. My final question for you, Jeff, really is and again, this is why we're going to have to definitely do a follow-up conversation. There's no question about it. Based on your experience at Vertex, we have a lot of listeners also that are biotech executives and leaders. What lessons would you offer to those leaders trying to build organizations capable of sustained innovation? I know one thing is I could say, read Jeff's paper. Read this. This is step one. Before you do anything else, read this. But if you were to kind of sum it up quickly at the end of this conversation, how would you sum it up to those biotech leaders?
Yeah, obviously there's no single simple answer, but a couple of things that I think are important, and there are many different ways that it can be done. One is, I think you do need a very clear, concise, and consistent strategy. If I had to say start with one thing, it's start with a really clear, concise, consistent strategy and stick to it and be deliberate about it. Two is you need a culture of collaboration. I just don't see how you do things in this industry without that culture of collaboration. To make a drug takes hundreds or thousands of people working for years and years together across many different functional areas, more so than even in many other industries. So creating this culture of collaboration, vigorous debate, vigorous discussion, cross-functional teamwork, but then everybody gets aligned and moves in the same direction. That cultural piece is just super important. And then the third piece is figure out a way to integrate internal and external innovation, because no matter how innovative you are, no matter how innovative Vertex is, we can't create more than 0.001% of the innovation in the industry, which means that we're going to have to rely on others to create some of the innovation, and we're going to need to access that innovation through collaboration and business development. And you have to have a strategy in which your external innovation and your internal innovation are highly integrated, and everybody's incented to do both together to make the best products. I think if you do those three things, you're going to win one way or the other. And there are many different flavors of it, but those three things have to be common to any successful long-term biotech. And remember, there have only been 5 biotechs that have created 4 or more drugs internally. 5 biotechs out of the thousands out there. So this is hard to do over and over again. But I think those three things are really helpful.
Absolutely. And again, Jeff, it was a real pleasure to have you on here. I know I'm going to get a lot of follow-ups on when can we get more from Jeff. But as a little teaser, there might be some written content coming out soon. And of course, we'll definitely try to plan a follow-up discussion with Jeff for a lot of the inbound that I'm going to get on other questions that many of you were hoping I'd ask. But Jeff, thank you so much. It was really an honor to get this opportunity to interview you. I'm really, really excited that Reshma connected us, and just keep up the incredible work. You know, everyone across the world is cheering Vertex on, and just please keep up the incredible work. It's just so exciting to see and to have this conversation.
Well, Chase, a pleasure to be here with you and tell the story. And it's really a pleasure to be interviewed by someone who's a physician who actually understands how this industry works. It makes for a really fun discussion. I look forward to following up.